As a lab specialist, the most two questions we hear: how long does the sample need? Where are the results? But no one asks what goes on behind the scene.
Today, we have a special sample called chorionic villus sampling. CVS is an important prenatal diagnostic test used to obtain a small sample of placental tissue, which contains the same genetic material as the fetus. The test is usually performed between weeks 10th and 13th of pregnancy by inserting a needle through the abdomen under ultrasound guidance to collect the sample. The collected tissue is then placed in a cell preservation medium and kept at room temperature until it arrives at the laboratory of chromosome analysis. Every answer begins with a single step upon arrival at the laboratory. Inside the laboratory, the journey starts with DNA extraction. The sample is carefully handled under controlled conditions, where every movement matters and every detail is protected from contamination. From placental cells, fetal DNA is gently isolated, separating it from proteins and unwanted impurities. What remains is pure genetic material. To check the purity and amount of DNA, measuring a single drop on the nanodrop machine gives you the exact concentration.
Inside the laboratory, multiple techniques work together to uncover genetic answers, among them PCR, that stands as one of the most powerful tools to amplification DNA. But before amplification begins, in the pre-PCR stage, the sample is carefully prepared under strict conditions to prevent contamination and preserve the integrity of the DNA. Only then the process begins. PCR allow lab specialists to generate millions of copies of DNA by using controlled temperature cycles, making genetic information easier to detect and analyze. But amplification is only the first step. To visualize this amplified DNA fragment, lab specialists turn to gel electrophoresis. In this step, DNA samples are carefully loaded into the gel matrix. An electrical current is applied, causing DNA fragments to move through the gel based on their charge and size. Smaller fragments travel faster, while larger ones move more slowly. What appears here is a distance pattern of bands. Each band represented DNA telling a precise molecular story.
But sometimes patterns are not enough. To understand the exact genetic code, scientists go even deeper. Through DNA sequencing, the precise order of nucleotides is revealed. Every base, every letter is carefully read and recorded. On the screen, genetic data appears as a complex pattern, a sequence that holds critical information about potential mutation. By analyzing these sequences, specialists can detect even the smallest genetic change that may impact a life before it even begins. And finally, the full picture comes into view. Through karyotyping, chromosomes are visualized and arranged, relieving their number and structure with clarity. The most common finding that this technique can reveal is Down syndrome, where the X chromosome in group 21 is clearly visible. From the single sample to a complete genetic story, this is the power of the laboratory.